Now that we have understood the steps in biofilm formation, let us move to a topic called as multi-species biofilms. When biofilms had been studied and are still being studied, it is most of the times that the studies about formation and understanding the complexity of the architecture, understanding the cell-to-cell communication between the organisms, most of these studies have happened with monospecies biofilms, and therefore most of our understanding is based on the biofilms formed by pure cultures. However, in nature, biofilms are usually multi-species biofilms. In nature, I mean to say in the environment as well as in the human body. For example, most of the biofilm based infections that happen in human body are also multi-species infections. In multi-species biofilms, we find that there is always a mixed population of organisms, and moreover, the interaction between the organisms that form these multi-species biofilms largely affects the structure and the functioning of the biofilm. What are the various types of interactions that happen between the organisms in a multi-species biofilms? One of the interaction is the quorum sensing that happens between different species. It is observed that interspecies communication that is mediated by autoinducers and take the pathway AI1 type 2 pathway leads to communication and therefore leads to co-aggregation of microorganisms in the biofilms, and this communication leads to the formation of more thicker and more denser biofilms. Communication or interspecies interaction between the microorganisms can also be in the form of cooperation or can be in the form of competition. Cooperation can be when the two microorganisms in the biofilm form more sturdier biofilms, or it can also be in the form of nutrient give and take. Microorganisms in the biofilm, multi-species biofilm, can also cooperate in a fashion, for example, when the microorganisms that are aerobic are present on the surface of the biofilm and they consume the oxygen and therefore allow less oxygen to diffuse inside and allow the anaerobic organisms within the biofilm to thrive better. Competition between the microorganisms can be in such a way where one of the organism secretes substances that will prevent the proliferation of another organism. Some of the beneficial phenotypes that are expressed by microorganisms in multi-species biofilms are listed here. Mixed biofilms are thicker and sturdier because of the co-aggregation and therefore have an enhanced biomass. Metabolic cooperation is easier due to the physical proximity and can allow succession in nutrient utilization, or it might also allow access to complex nutrients that are used by the organisms successively. This phenomena is also called as metabolic commensalism. One classical example is oral biofilms, where it has been found that oral bacteria rely on interspecies interactions when forming mixed biofilms. Several studies have evaluated the antibiotic susceptibility of multi-species versus monospecies biofilms. It has been observed that microorganisms in multi-species biofilms have an increased resistance to antibiotics or to host responses as compared to the monospecies biofilms. One reason could be that the EPS matrix of mixed biofilms is more impermeable to antimicrobials. Here are some pictures of biofilms that are detrimental. Biofilms can hinder industrial processes by causing biofouling in cooling towers and pipelines. As you can see, thick biofilms formed in pipelines in one of the pictures there. They can also pose a risk to public health, and they can also be medically significant. One of the detrimental biofilms that you can see here is the oral biofilm formed by bacteria like Streptococcus mutans that lead to tooth decay. Detrimental biofilms can also be formed on implants and prosthetic devices. The electron micrograph here is a picture of biofilm that is formed on an implant. These are, as I had earlier mentioned, are very significant because it is very difficult to treat such implant based infections with antibiotics, as the antibiotic resistance of these organisms is much higher than that of the planktonic cells. The other pictures that you can see here are pictures of biofouling that is caused on ship hulls by microorganisms including algae.